بسم الله الرحمن الرحيم.
Dear students, today we will go through lecture 14 of organic based industries. The topic of today's lecture is nanotechnology and drug delivery. Nanotechnology is the manipulation of matter on an atomic, molecular and supramolecular scale, involving the design, production, characterization and application of different nanoscale materials in several key areas, providing novel technological advances, mainly in the field of medicine, so called nanomedicine. Nanotechnology for drug vectorization provides for new and more specific drug targeting and delivery platforms that can reduce toxicity and other side effects and also maintain or improve the therapeutic index. Key concepts that we will go through in today's lecture include polymer for drug delivery systems before and after nanotechnology, timeline of nanotechnology, liposomes, transdermal microarray patch, nanoparticle mimicking red blood cells, and lab on chip. Why drug delivery is a crucial part to pharmaceuticals and how nanotechnology is advancing drug delivery. The importance of nanotechnology in drug delivery is in the concept and ability to manipulate molecules and supramolecular structures for producing devices with programmed functions. For a drug to be successful, it must satisfy pharmacokinetic and pharmacodynamic requirements. Pharmacokinetic requirements are that it must be absorbed, distributed, metabolized, excreted sufficiently, and non-toxic. Pharmacodynamic requirements are that the drug must have sufficient efficacy, that is, ability to carry out the desired effect, and selectivity. When a drug enters the body, there are certain biological barriers that the drug molecule must first pass to get to its target organ. For example, when a drug is taken orally, it must pass barriers in the stomach, the small intestine, the blood, and the liver before it reaches the target organs. Barriers which must be overcome in these regions are cell membranes, metabolic enzymes, efflux transporters, and binding proteins. Needless to say, there are many criteria which a drug must possess in order to reach its target region. About 70% of drugs today are taken orally. Most pharmaceutical companies aim to develop an oral dosage form of their drug with a dosing regime of once per day. Drugs of this type have reasonable manufacturing and storage costs and high patient compliance. Drug delivery for drug administered orally usually involves encapsulating the drug using polymers. Liposomes are the main drug delivery systems used today in oral administrations. Drug administered by parenteral routes, those which do not encounter stomach or gastrointestinal tract, must also cross certain barriers. However, the basis of drug release from drug delivery system is the same for all routes. Drug release is from diffusion, degradation, swelling, and affinity based mechanisms. To appreciate the true meaning of nanotechnology in drug delivery, it may be beneficial to classify drug delivery systems based on the time period representing before and after the nanotechnology revolution. Most common types of administration are oral, topical through skin, transmucosal including nasal, buccal, vaginal, oral or rectal, and fourth is inhalation. Liposomes, polymeric micelles, nanoparticles, dendrimers, and nanocrystals are a remaining portion of conventional drug delivery systems that happen to be on nanoscale. Nanotechnology and its effects on drug delivery refers more correctly to the current drug delivery systems, which include microchips, microneedle based transdermal therapeutic systems, layer by layer assembled systems, and various microparticles produced by inkjet technology. The main problem with the trivial methods are low drug loading capacity, low loading efficiency, and poor ability to control the size distribution. Utilizing nanotechnologies such as nano patterning could allow manufacturing of nano or microparticles with high loading efficiency and high homogeneous particle sizes. The pharmaceutical industry has been slow to utilize the new drug delivery systems if they include components also called excipients that are not generally regarded as safe. Going through clinical studies for FDA approval of a new chemical entity is a long and costly process. There is resistance in industry to adding any untested materials that may require seeking approval. Nanotechnology for drug delivery will mature faster and become more useful if it is appreciated that the real potential of nanotechnology in drug delivery is based on utilization of nano or micro fabrication and manufacturing rather than on dealing with delivery systems in nano or micro scale. To describe what nanotechnology can do to manufacture nano or micro drug delivery systems, one can use manufacturing of nano or micro particles or capsules as an example. Imagine that the current soft gelatin capsules which are in centimeter scale, are manufactured at nano or micro scale. The following are among the important technological advantages of nanoparticle as drug carriers. High stability, that is long shelf life. High carrier capacity, that is many drug molecules can be incorporated in particle matrix. Feasibility of incorporation of both hydrophilic and hydrophobic substances. And last, feasibility of various routes of administration, including oral administration and inhalation. These carriers can also be designed to enable controlled or sustained drug release from the matrix. An example of where drug delivery by a nanostructure is required. Small interfering RNA is a potential new universal drug for treatment of a variety of human diseases, but efficient delivery into diseased cells remain a major challenge. Polymeric nanocarriers containing the drug may solve the problem. Within the body, naked small interfering RNA is degraded by enzyme. To avoid this, siRNAs are incorporated in nanoparticles able to reach target cells intact. Microencapsulation technology has been used to surround nanoparticles in a biodegradable coat for sustained release delivery. After release, the nanoparticles bind to receptors in cell membrane and are subsequently transported into the cell. Inside the cytoplasm, the nanoparticle matrix dissolves and drug is released. Liposomes are nano-sized artificial vesicles of spherical shape that can be produced from natural phospholipids and cholesterol. The properties of liposomes, in addition to general properties of surfactants, those make them useful for different applications are structural stability on dilution, varying permeability of the bilayer to different molecules, and ability to entrap both water-soluble and insoluble substances and deliver them into desired environment. The size, lamellarity, unilamellar or multilamellar, and lipid composition of bilayers influence many of important properties like the fluidity, permeability, stability, and structure. These can be controlled and customized to serve specific needs. The properties are also influenced by external parameters like temperature, ionic strength, and presence of certain molecules nearby. Liposome is extensively studied for encapsulation of drugs. When lipid self-assemble to liposomes, water-soluble drugs will be trapped inside the liposomal cavity. Fat-soluble drugs are incorporated within phospholipid bilayer. The lipid bilayer of liposome can fuse with other bilayers, for example cell membrane, thus delivering the liposome contents. Liposomal formulations are the first nanopharmaceuticals introduced to market. These liposomes are called as stealth liposomes with size less than 200 nanometer, which are long circulation with hydrophilic polyethylene glycol surface. These long circulation liposomes found to target the tumor tissue by a mechanism known as enhanced permeation and retention (EPR). Hence, liposomal formulation of doxorubicin considerably reduced the cardiotoxicity of drug. Many liposomal products are under various phases of clinical trials. Liposomes are currently investigated for a variety of additional therapeutic agents, for example, anti-cancer drugs, antibiotics, and biologics such as antisense oligonucleotides or DNA.
A microarray patch technology is being developed for the transdermal delivery of large molecule drugs without the use of injections. The patch is in a band-aid format, and the surface of the patch is structured with polymer microneedles from which the drug is delivered. The drugs can be attached to the external surface of the polymer microneedles, integrated into the polymer, or both. When the patch is applied, the microneedles cross the stratum corneum and penetrate into the epidermis. The microneedles do not penetrate deep enough to enter blood capillaries or nerves, hence the delivery is non-invasive and pain-free. The drugs for delivery are present in a nanostructured form, facilitating uptake into the body. The microneedles are made of a polymer that is biocompatible and biodegradable. This reduces the risk of trauma to the skin and infection. The microarray patches have been designed for the delivery of peptides, proteins, hormones, vaccines, and skin repair agents. The use of microarray patches will enable a wide range of medications to be effectively delivered to humans in a safe and non-invasive manner.
In the short clip on slide, we see particles which measure 6 micrometers in length passing through membrane half their width. They mimic red blood cell shape and consistency in that they are flexible and foldable. Basically, in this experiment, it was proven that more flexible the molecules are, the longer they last in circulatory system. It was also seen that molecules with different flexibility ended up in different organs. This may prove to be a very beneficial advancement in drug delivery. De Simone, the chemist who worked on this, says with regard to these findings that hopefully application for the delivery of cancer drug could be in early clinical trial stages within few years. Delivering cancer drugs with nanoparticles, MIT and Harvard researchers have turned their attention to cardiovascular disease, designing new particles that can cling to damaged artery walls and slowly release medicine. The particles dubbed Nanobursts are coated with tiny protein fragments that allow them to stick to damaged arterial walls. Once stuck, they can release drugs such as Taxol, which inhibits cell division and helps prevent growth of scar tissues that can clog arteries. A lab on chip (LOC) is a device that integrates one or several laboratory functions on a single integrated circuit, commonly called a chip, of only millimeter to a few square centimeter to achieve automation and high throughput screening. LOCs can handle extremely small fluid volumes down to less than picoliter. Lab on a chip devices are a subset of microelectromechanical systems known as MEMS and sometimes called micro total analysis systems, micro TAS. Lab on chip may use microfluid physics, manipulation and study of minute amount of fluids. However, strictly regarded, lab on chip indicates generally the scaling of single or multiple lab processes down to chip format, whereas microtas is dedicated to integration of total sequence of lab processes to perform chemical analysis. The term lab on a chip was introduced when it turned out that microtas technologies were applicable for more than only analysis purpose. About 15 years ago, MIT professors Robert Langer and Michael Sima had the idea to develop a programmable, wirelessly controlled microchip that would deliver drugs after implantation in a patient's body to administer daily doses of a drug normally given by injection. To circumvent the barriers encountered by macromolecules at gastrointestinal mucosa, sufficient therapeutic macromolecules must be delivered in close proximity to cells. Nanotechnology protects capsules from counterfeiting and diversion. Employing nanotechnology based encryption onto pharmaceutical capsules before they are filled adds overt, covert, and forensic level protection. Delivering therapeutics to mucosal tissues such as nasal and gastrointestinal tracts is highly desirable due to ease of access and dense vasculature. However, the mucus layer effectively captures and removes most therapeutic macromolecules and devices. Nano-engineered microparticles (NEMPs) adhere through the mucus layer, exhibiting up to 1,000 times the pull-off force of an unmodified microsphere and showing greater adhesion than some chemically targeting means. Nanotopography improves device adhesion in vivo, increasing retention time up to 10-fold over unmodified devices. Physical interaction of nano-engineered surfaces with subcellular structures producing a robust and versatile cytoadhesive for drug delivery. These nanoscale adhesive mechanisms are also relevant to fields such as tissue engineering and wound healing because they likely affect stem cell differentiation, cell remolding, migration, etc.